In this video, we're going to look at the membrane skeleton. And to study membrane structure, you usually use erythrocytes. Erythrocytes are cells that generally, like organelles, hardly does metabolism. You can just think of it as just a membrane bag that holds hemoglobin. And to just get the membrane, you do osmotic lysis, which you make the cell contents leak out, and you're left with just the membrane, which are known as erythrocyte ghosts. And erythrocytes have this biconcave disc-like shape, and this disc actually moves along the membrane. It isn't fixed. It will roll across the skeleton. And this rolling is due to a network of proteins that we call the membrane skeleton. This allows erythrocytes to be fluid and to change shape. For example, when moving through capillaries, you have to squeeze, and the skeleton and disc-like shape allows it to do that. Now let's look at the proteins that make up the membrane skeleton. And the main protein is called Spectrin. And it's called Spectrin because it was found in the erythrocyte ghosts. Now this is roughly 75% of the erythrocyte membrane skeleton. It's 280 kilodaltons of alpha subunits, as we see here. Alpha helixes, and you have two different subunits, the alpha chain and the beta chain. And these have 106 residues, triple alpha helix coiled. So here we're looking at the triple alpha helix coiled, and these will form into the alpha chain and into the beta chain. And these two chains will wrap around each other to get a dimer that's roughly 1,000 angstroms long. You then have two of these heterodimers. So it's a heterodimer because it's made out of an A and B chain, and they'll associate head-to-head to form an AB tetramer. So two of these will come together to form a tetramer. And Spectrin looks a lot like the protein dystrophin, which is the muscle protein that is defective if you have muscular dystrophy. Now, continue looking at this membrane skeleton. You roughly have 100,000 of these Spectrin tetramers per cell, and these are cross-linked at both ends by attaching to other cytoskeleton proteins. So here we have a diagram of that. You can see Spectrin, and here at the cross we are cross-linked here, cross-linked here. Here's what it actually looks like, and you can see your Spectrin little skeletons right there. And this is dense and irregular meshwork, so there's not really a pattern here. If you are deficient or your Spectrin doesn't quite work, you have hereditary spherocytosis. And that's because your erythrocytes, instead of being this concave shape, are now perfect spheres. That makes them fragile and inflexible. And you'll often have anemia because your erythrocytes will break. They'll be destroyed by the spleen. Your hemoglobin can't get to where it needs to go. Therefore, you cannot get oxygen to your tissues. Now, one of the proteins that Spectrin associates with is called Ankyrin. And you can tell by the name, Ankyrin serves as an anchor. So Ankyrin binds to an integral membrane ion channel. And since Spectrin binds to Ankyrin, this will actually anchor the skeleton to the membrane so it doesn't float away. You also have Spectrin linked proteins and Ankyrin linked proteins in a variety of tissues. So they're proteins that look like Spectrin and Ankyrin. Now, Ankyrin is 798 N-terminal segment, contains 2433 of these Ankyrin repeats. So each one of these things is what we determine to be an Ankyrin repeat. They're two short anti-parallel alpha helixes followed by a long loop. So you can see alpha helix, alpha helix, alpha helix, alpha helix, alpha helix, alpha helix. And these form this concave surface. This is where Spectrin and integral membrane proteins are thought to bind, as we can see by this lovely picture taken at UT Southwestern in Dallas. Now, if we look at this cartoon of the membrane skeleton, we can start to understand why some proteins are free to move throughout the membrane and some aren't. So some proteins, such as B, are trapped in what are called fences. So this Spectrin cytoskeleton forms these fences where this protein cannot leave. Or they are attached to the skeleton, such as A, since they can't move at all. Others, such as protein C, are not attached. They're not in this fence, and so it's free to move. This is called the gates and fences model. So gates, fences. If you destroy the cytoskeleton, you will see greater protein diffusion, which is helping to confirm this model of the membrane skeleton. Now, when we think of the membrane, the different sides, the sides that face the cytoplasm and the sides that face the extracellular space, have different amounts and types of membranes in them. And this is shown in this graph. The inner leaflet is pointing towards the cytoplasm. The outer leaflet is pointing to the outside of the cell. Now, to make membrane lipids, the proteins that do that are mostly integral membrane proteins, and they're found in the ER. Now, prokaryotes, they do it a little bit differently, but this is the same idea. Their integral membrane proteins are in the plasma membrane. Now, when you make a lipid, it's going to be inside the cell, and you have to have some way of transferring lipids from the inner leaflet to the outer leaflet. Usually this is energetically unfavorable because the head group of the lipid has to pass through the hydrophobic part of the membrane. Not favorable. So we have a couple of enzymes that will change lipids from different sides of the membrane. One is footbase. So it will take specific phospholipids and move them from the different sides of the leaflet. It basically hides the charged head group, so it's not energetically a bad process. This is facilitated diffusion. While phospholipid translocase transports specific phospholipids using ATP hydrolysis, and this is active diffusion. And now, the lipids on the outer leaflet can be a signal. For example, if you have phosphatidylserine showing on the outside of a cell, this is a signal to start the process for blood clotting. So this is one way that different cells can talk to each other. Because if you look at the inner leaflet, phosphatidylserine is only found in the inner leaflet. So if any is showing on the outer leaflet, this is a signal to say, hey, start blood clotting. And normally it's not found there. Now, when we talk about the organization of the membrane, we have to realize that actually some parts of the membranes are organized. So in the last video, I talked about the fluid mosaic model, which is random organization, and that's not quite the whole picture. There is some layers of organization. In fact, the plasma membrane of eukaryotic cells, not elves, have two or more distinct, not distant, can't type, domains with different functions. Basically, the plasma membrane had different lipids and proteins for these different domains. And actually, small parts of the plasma membrane are often segregated to form microdomains, smaller domains, which you have enriched in certain lipids and or proteins. And a common type of these microdomains or feature of the membrane that you see are called lipid rafts. Now, these lipid rafts have glycosphingolipids and cholesterols, as shown here, all tightly packed together. And this cholesterol is kind of shoving your lipids together to make it a more ordered or crystal-like arrangement. And certain proteins, shown here in blue, will actually associate with these rafts, and they're usually GPI linked proteins. Now, these types of lipid rafts are probably in all cell types, and they are probably for complex intracellular signaling. these lipid rafts. And interestingly enough, influenza, measles, Ebola, and HIV all localize to lipid rafts as one way to get into your cell. They will bind to these lipid rafts. And that's it for this video. I will see you in the next one.